Hello everyone and thank you for coming. My name is Jade. I am a PhD student at the University of Hong Kong, and today I will tell you about my research on the effects of developmental temperature on the locomotory innate and learned behavior to olfactory cues in the zebrafish Dinuario. I will start with reminding that temperature is a primary factor of influence in the life of aquatic ectotherms, which is important to keep in mind nowadays because we're currently experiencing global warming and will continue to do so in the near future. This global change can impact in many ways aquatic animals, notably when we speak about behaviour. It is important to understand what the effects of near-future temperature changes on the behaviour of aquatic animals will be, because it can have repercussions on population dynamics through differential survival. So, in our study, we are investigating the effects of developmental temperature on the behaviour and the learning abilities in the larval zebrafish, which is a neurological model species. In order to test whether elevated temperature influences swimming behaviour, we have exposed zebrafish from embryo stage and for 7 days to either control temperature, which was 28 degrees, as you can see on the left, or elevated temperature, which was 30 degrees, as you can see on the right. At 7 days post-fertilization, we started our experiment by individually video recording routine swimming activity for 12 minutes. In order to test whether elevated temperature affects the fish response to olfactory cues, we then exposed the larvae of both temperature groups to either one of the three cues. There was a water control that you can see on the right in both temperature groups. There also was a catfish cue that you can see in the middle of the figures, which is likely encountered in the zebrafish's natural environment, and it was shown that larval zebrafish do not change their swimming behaviour in the presence of that cue, so we call it neutral. Finally, there was a conspecifics alarm cue that you can see on the left of the figures, which was produced with conspecifics of the same cohort. Exposure to conspecifics alarm cue produced a freezing response in many fishes. After exposures to either of those three cues, the larvae swimming activity was recorded again for 12 minutes. We also conducted conditioning on some larvae to test for their learning abilities and to see whether elevated temperatures could affect it. In both temperature groups, conditioning consisted in exposing the larvae to a mixture of a neutral cue, in our case the catfish cue, and the conspecifics alarm cue, at 6 days post-fertilization for 40 minutes. Then, 20 hours later, the larvae were recorded 12 minutes before and after exposure to either the water control or the catfish cue alone. We aimed to see whether the fish would then have a freezing behavioural response to the catfish cue, which would mean the neutral cue was assimilated to a threat, thanks to conditioning. This protocol proved effective in older larvae reared at controlled temperature in a previous study. From these assays, we calculated the total distance swam throughout the experiments, before and after cue exposure for each larvae. And what we first found was that there was an effect of temperature on baseline routine swimming activity, as you can see on this figure, displaying the total distance swam according to the rearing temperature. Larvae reared at 30 degrees swam less distance than those were at 28 degrees during baseline activity. This result is in accordance with the thermal performance curve dogma, as zebrafish's optimum temperature was described to be 27 to 28 degrees, and our elevated temperature treatment is 2 degrees Celsius past it. What we found next was a little surprising because there was no alteration of the total distance swam after exposure to any cue, including the conspecifics alarm cue, as you can see on the top figure, displaying total distance swam across time for each of the three cue groups, here from the control temperature cohort as an example. So the black line represents larvae exposed to the controlled water, the orange line represents larvae exposed to the neutral catfish cue, and the blue line represents larvae exposed to the conspecifics alarm cue group. The red line shows when the exposure cue was introduced. We also found no effect of temperature on that response, so it may be that the larvae were too young to respond to that specific cue, as it was shown they can detect it and respond to it from 12 days post-fertilization onwards. It may also be that the cue prepared was not concentrated enough to trigger the behavior in this aged larvae, as another study found that 6 days post-fertilization larvae could respond to a different and more concentrated alarm cue called Schreckstoff. Finally, regarding conditioning, although we did not find any significant difference in total distance swam indicative of learning in any temperature group, we seem to detect a slight increase in swimming for conditioned larvae that were exposed to the catfish cue, as you can see on the bottom graph displaying distance swam over time, for both the water control exposed and the catfish cue exposed larvae. Here from the elevated temperature group as an example, we've also found the same result in the control group. So this part needs more investigating to see whether learning did occur or not in 7 days post-fertilization larvae, as well as tests and other metrics in the future. Thank you for listening.